In this video, we'll begin our discussion of proteins. Proteins are complex structures constructed of amino acids. Proteins are macromolecules made of monomers called amino acids. So what are proteins? They're important for a healthy diet. If you lack proteins, this could lead to potential health problems. They contribute to the structure of the human body. So a lot of proteins, for example, are fibrous, and they're important for giving strength to your skin, to your hair, to your nails, for example. Proteins are also tiny machines that carry out many cellular functions. So we'll talk a lot about proteins in this class. Each amino acid has essentially this a basic structure. And we can start with the central carbon atom, which you can see here in the diagram to my side. Attached to the central carbon atom are three groups that are the same for all amino acids. There's an amino group, which is NH3+. There's a carboxyl group, which is COOH. There's always a hydrogen. And then finally, each amino acid has a specific side chain, also known as an R group. There's an example just above me, which is alanine, which has, again, you can see the central carbon, you can see the carboxyl group, you can see the amino group, the hydrogen, and then the side chain or R group for alanine is CH3. So proteins in your body are made from 20 different amino acids. Each type of amino acid is unique because it has a specific side chain or R group. Some of these R groups are nonpolar, and an example of that is shown here. We have alanine, which has its side chain or R group being CH4. Now remember that carbon and hydrogen, although they don't have exactly the same electronegativity values, they're similar. Carbon is ever so slightly more electronegative than hydrogen, but they're close enough, and so we consider the covalent bonds between carbon and hydrogen to be nonpolar. Therefore, this whole R group on alanine is nonpolar. And that's important because the chemical properties of the R groups or side chains is very important for the overall chemical properties of the protein. And we'll discuss that further in other videos. Now, some R groups or side chains are polar. And an example of that is shown here. We have asparagine. Its side group, you can see, has an oxygen. Oxygen is more electronegative than carbon, so that's going to be a polar, it's actually a polar double covalent bond. And then if you look, we have nitrogen. It's more electronegative than carbon, so that's a polar bond there between nitrogen and carbon. And also nitrogen is more electronegative than hydrogen. So you can see that nitrogen is covalently bonded to hydrogen here in asparagine, and so that would also be a polar covalent bond. Therefore, this whole side chain or R group for asparagine is in fact polar, and that again has properties that are important for the overall functioning of the protein, which we'll talk about more in other videos. Now, I haven't shown it here just to keep things simple, but again, there's 20 different amino acids. Each one is unique, and there are some also that have a negative charge and others that have a positive charge. And that's, of course, also important for the properties of the protein. Okay, so how do these, so I've mentioned that the subunit of a protein is called an amino acid. So proteins are essentially chains of individual amino acids linked together by covalent bonds, and they're formed by dehydration synthesis. The term polypeptide is used to refer to a polymer consisting of many amino acids bonded together in a chain. And we often use the term protein to refer to such a chain when it's longer than 100 amino acids. Now, this number isn't set in stone, but that's just sort of a rough dividing point in which we start to talk about a polypeptide as being a protein. And part of that has to do with the fact that as the polypeptide gets longer, it starts to have a unique three-dimensional shape. And so that's something we'll talk about in another video. Let's take a look at how this dehydration synthesis works. We have here an example. We have alanine and valine. You can see that during dehydration synthesis, we should have water coming off, and you can see H2 coming off, H2O coming off. The oxygen comes from the alanine, from the carboxyl group, and the two hydrogens come from the amino group of valine. And after this dehydration synthesis process happens, you now have a covalent bond between the alanine and the valine. And we're essentially starting the formation of a chain. And you can see right above me just an example sequence of amino acids. So basically you could have repeated dehydration synthesis reactions, and this chain could get longer and longer. In fact, it could be hundreds or even thousands of amino acids in length. So that's it for this video. It's just a quick introduction to proteins. One of the key points is that proteins are polymers. They're made out of monomers, and each monomer is an amino acid. There are 20 of these amino acids, each with unique properties due to their unique side chains, which are also called R groups. Thanks a lot for watching.